Welcome back, everybody. This is MMBS-111 Introductory Microbiology. This is part 1 of mini lecture 5.3. We're going to be discussing viral replication and the viral life cycle. There are 4 learning objectives for this mini lecture. First, list the 5 steps of the life cycle of animal viruses. Second, define host range and tropisms. Third, define cytopathic effect and give an example. And finally, describe the mechanism and impact of persistent infections. This image is similar to the one in your text, and it quickly breaks down the steps or phases of the life cycle of animal viruses. So we have 6 steps. First is adsorption, and this is where the virus attaches to the host cell membrane and then is brought into the cell through endocytosis within a vacuole. Then we have steps 2 and 3 are penetration and encoding. So with penetration, the virus, after it attaches, is engulfed into the cell through endocytosis. Within that vesicle, it becomes uncoated, so the nucleic acid is then exposed. And then we have synthesis, which is step 4. And this is where all of the virus parts necessary for that active virus are formed. Step 5 involves the assembly of all of those parts into a complete and active virus. And then finally, the virus is released from the cell as a complete virion and is ready to infect another cell. This process can take anywhere from 8 to 36 hours depending on the type of virus. So it can happen rather quickly. So let's talk about some of the details of each of these steps. First, we have adsorption. And adsorption is very specific between the host cell and the virus. And this involves the range of the virus as well as the tropism. So a virus that is able to infect many species of organisms and many different cell types is said to have a broad host range, whereas a virus that is only able to infect certain cell types or certain species is said to have a limited host range. Each virus must link to a specific receptor on the surface of a cell. So any cells that lack those specific receptors will avoid absorption and invasion by that virus because the virus is not able to make that connection. Steps 2 and 3 are penetration and encoding, and this can happen in a number of different ways depending on the virus itself. So penetration can happen either directly through the fusion of the virus envelope with the cell membrane or through endocytosis. If the virus is a naked virus, then the capsid is going to connect directly with the cell membrane, and then it may inject its genome directly through the plasma membrane and into the cytoplasm. It's also possible that a naked virus could be engulfed through endocytosis. And endocytosis we sometimes term as cell eating. So the virus will connect through adsorption to the cell membrane, and then it'll be engulfed into the cell through a vesicle. And the vesicle and the virus's cell envelope will then fuse and release the capsid into the cytoplasm, where enzymes will break down the capsid and release the viral DNA into the cytoplasm. It's also possible that the envelope of the virus fuses with the cell membrane, and that fusion then opens the virus envelope into the inside of the cell where the capsid is released, and enzymes will then break down the capsid of the virus to release the viral genome into the cytoplasm so that we can begin synthesis. Depending on the type of nucleic acid that the virus possesses, that will determine where synthesis takes place. So if this is a DNA virus, the DNA is going to enter the host cell's nucleus, and that is where synthesis and replication and assembly will take place. If the virus is an RNA virus, then all of those processes will happen within the cytoplasm, with the exception of retroviruses. Because retroviruses possess RNA, but they bring DNA polymerase with them, they're going to reverse transcribe DNA from the RNA they brought in their capsid. And then once that happens, the DNA will enter the nucleus of the host cell, and then those processes will continue within the nucleus. This table is from your textbook, and it just shows you the transcription and translation modes that are available to viruses as they enter the host cell, depending on what type of nucleic acid they have. The last two steps of the animal cell virus life cycle are assembly and release. And assembly, we're just talking about putting all of those manufactured parts together. Once the virus is assembled, then the virus is going to be released from the cell. And the number of viruses that can be released depends on the type and size of the virus itself, as well as the size and health of the host cell. So small viruses, we're going to end up with a larger number of active virions than with larger viruses. However, we do end up with thousands of active virions, which means that infection and disease can spread rapidly in the host body. This image shows you a diagram and an actual image of the release step from the viral life cycle. You can see that the process is very similar, sort of backwards to how the virus enters the cell. So once everything is assembled, then the virus is going to come in contact with the membrane, whether it's the cell membrane or the nuclear membrane. It's going to be enclosed in that membrane, and then it will be released to make contact with new cells and spread that infection. Image B shows you what this looks like in actual cell, and you can see that pleomorphism that we talked about previously, how with the virus being surrounded by the cell membrane, it assumes different shapes. So viruses do quite a bit of damage to host cells, and we call this damage cytopathic effects. And you can see this damage as you look at the cells under the microscope. A number of things can happen to host cells through viral infection. We can have just an overall change in the cell's size and shape. Sometimes viral infection will produce inclusion bodies within the cytoplasm or the nucleus of the cell, depending on where viral replication and assembly happened. And these inclusion bodies are typically either compacted masses of virus or damaged cell organelles and other parts that have been contained in these inclusion bodies. Syncytia can also occur where multiple host cells fuse to one another. So we have this very large host cell with multiple nuclei contained within the cell. Most host cells do not survive this kind of damage and eventually die after viral infection. Not all viruses immediately leave their host cells after replication and assembly. Some viruses remain within the cells of their host for weeks or for the entire life of their host. They can remain in the cytoplasm, or they can incorporate into the DNA of their host cell. And this results in latent infections. So things like the herpes virus, these latent infections can be activated because the virus remains within the host cell, and when there is a stress on the host body or the cell, that virus can be activated and cause a latent infection like a cold sore or shingles. And that is the end of part 1. Make sure that you can answer these questions accurately and go back and review the learning objectives to make sure that you understand those. Hopefully this video was helpful.